Welcome everybody to our December edition of our MycoTalks series. My name is Gordon Brown. I am at the MRC Centre for Medical Mycology at the University of Exeter. And my co-chair today is Joe Heitman from Duke University. We have two fantastic talks for you today. The first is from Professor Judith Berman, who's going to be telling us about how we can learn from pathogenic yeast about tolerating changes. And the second is from Michail Lionakis, who's going to be talking to us about aberrant type 1 immunity and mucosal fungal susceptibility. Now, for those of you who haven't joined us before, the purpose of this seminar series is really to provide the community with an open access platform in which we can all share in the wonderful diversity of research that contributes to the field of medical mycology. So the structure of this is that we have two talks every session, and we each try to aim that we have different talks around different subject areas in each one of these meetings. And each of the talks will last for approximately 30 minutes. And we'll have the talks one after the other. And at the end of the talks, we'll have a joint question and answer session. And so for this, I would really ask you to submit your questions in the Q&A. You'll see at the bottom of your screens. We're not monitoring the chat function, so please do submit them there. And please also just remember to put the name of the speaker to whom your question is addressed at the beginning of the line. And then the way this works is that Joe and I will be looking at the list of questions that are submitted, and we will then do a tag team event at the end of the two talks, presenting your questions to the speakers. Now, these questions will be selected, they will be grouped potentially by us, and they will be selected out of order to which they're submitted, and we will do our very best to get through all the questions that you submit in the time allowed. So we have about half an hour for questions at the end. And if you know of people who've missed these talks or you want to come back and watch them again, we record all of these talks, and they are available on our YouTube channel. And you can see at the bottom of your screen there the link to that location where you can find these talks. Just to remind you that you have to register for every one of these MycoTalks for each month. And again, our web address linked at the bottom there will direct you to the right place and where you can register for these talks, and to highlight our upcoming talks in January and in February. And then finally, just to thank the team behind this series who make this all very easy and make it happen every month. And that's Jules, Lorna, and Alberta. And with that, I've finished my introduction, and I will hand over to Joe, who will introduce Judy. Joe.
Well, it's really an honor and a pleasure to be here today and to introduce my friend and colleague, Judith Berman. Judy was at the University of Minnesota for the first part of her career, and then 8 years ago, moved to Tel Aviv University, where she's a professor. Her key discovery is that aneuploidy leads to drug tolerance and drug resistance, and the mechanisms by which aneuploidy exerts these effects. And of her many honors, I want to highlight two. She was elected a member of EMBO last year, and then this year she was awarded an ERC Synergy Grant with Marcus Ralzer in Berlin for their studies on drug tolerance mechanisms. Welcome, Judy. We're really excited about your MycoTalk today.
Thank you very much, Joe. I need to share the screen. I think it's there. Okay, is that working? Can someone let me know if you can see it?
That looks terrific.
Okay, great. So thank you very, very much. And it's really a pleasure and an honor to be here. This series is really quite impressive, and I'm hoping that I can stand up to the level that it's been set at. And I really look forward to the discussion afterwards. I think the discussions have been incredibly robust. I'm going to talk today about antifungal drug resistance and tolerance, and trying to help distinguish between them and show some of the things we think about their mechanisms. Oops, let's see here. So this is just, I feel like I'm preaching to the choir here, the impact of fungal infections, but I think it's important to remember that fungal infections are a very big problem, and the number of fungal infections is approaching that of some of the very big three: malaria, TB, HIV, AIDS. And with cryptococcosis in sub-Saharan Africa, the mortality is very high, and with invasive candidiasis, as well as with invasive aspergillosis and pneumocystis, the mortality is extremely high. So mortality percentages, this shows the highest that has been reported, but in general for candidiasis, which I'll be focusing on today, it can easily be in the 30 to 50% range, which is really very high. And this is in patients for candidiasis that are being treated in Western hospitals with the drugs that we have available. So we really need to do better, and the question is how to attack that. So one of the things that you see in lots of reviews is that drug resistance is increasing, and the question is why. And one of the answers is that there's an increase in prevalence of non-albicans species, which are species that either have higher levels of resistance to start with, or more frequently are more able to adapt to the antifungal drugs that we give to patients, and therefore to persevere or to persist in the host and cause a series of problems over longer time. This is just showing the distribution of the second most the two most prevalent fungal Candida species that are causing infections around the world. And you'll see that in North America, in much of Europe and Australia, it's usually Candida albicans with Candida glabrata, whereas in South America, South Africa, and Spain and China, it will be Candida albicans with parapsilosis. And in very tropical areas, you can see a lot of Candida tropicalis in addition to albicans. Another increase is the appearance of new, entirely new, intrinsically resistant and adaptable species. And the poster child for that is Candida auris, with numbers of infections rising, especially in hospitals and nursing homes. This is many of the isolates are intrinsically resistant to fluconazole, and they have the ability to develop resistance to the other two major classes of antifungal drugs. But Candida auris is still not causing very large numbers of infections. It's something that's fairly rare, but it does spread and causes lots of concern because of that spread. In addition, there's a concern for fungal infections with resistant isolates from the environment, and in particular from agricultural sources. So in agriculture, they use azole antifungals in amounts far higher than we use in the clinic. About 300-fold higher is the number I read recently. And the spread, then you can have a spread of those resistant environmental isolates to the population that resides nearby, and particularly in Europe, where the population centers are very close to agricultural areas, this has become a major concern. For Candida species, which I'm going to focus on today, these are commensals of humans, and this is a less likely route of resistance. But I don't think anyone's ruled out the idea that if we're eating foods that have these azole antifungals in them, that it may not influence the adaptability of our microbiome as it suffers more exposure to these stresses. However, when surveys have been done of Candida species, what is found is that the number of resistant isolates in the hospital is not increasing. And so this is the case, but we still have this very high rate of treatment failures. The number of Candida infections is the number of resistant Candida infections is between 1% and 5%, depending on the report, of all isolates. But the number of failures is far higher than that. And so susceptible isolates must be able to continue to cause disease despite their being treated with the drugs that we have available. So what are the reasons for treatment failure? Well, the classic one is host factors, and immune status is a major one that we know is important. But in addition to that, there can be drug properties such as the dose of the antifungal that we're giving or the pharmacokinetics of that drug and whether it gets to the site of infection, as well as interactions with other drugs the patient may be taking and the static versus societal effect of the drug. But I'm going to focus today on the fungal properties. And while in general, there's been a focus only on resistance, and that's probably because we use the bacterial paradigm that's out there and very classic, tolerance, I think, is an important thing to take into consideration, and I want to make that argument today. So I'm going to focus on these two things. So first, we need to define the difference between resistance and tolerance because they're often used very interchangeably, these two words, in the literature. And in fact, a problem with these words is that we use them in common vernacular as well. And I think when we're talking about these in the scientific context, we need to be very careful to talk about them separately. So the clinical definition of drug resistance is one that is important for clinical application, of course, and it, of course, uses the minimal inhibitory concentration, or MIC, and resistance is when that MIC is above a clinical breakpoint that's been empirically determined such that it's a breakpoint above which, if the MIC is higher than that, then therapeutic failure is more likely. The research definition that we use in the lab is that the MIC of a resistant strain is higher than that of a susceptible isolate, which is usually a control or a reference strain. So for example, here I'm showing you disc diffusion assays. There's drug in the disc, in this case fluconazole, and there was a lawn of cells plated across the whole plate. And the drug diffuses into the plate and generates a zone of inhibition, such that here the zone of inhibition is of a particular size that can be measured from its radius. And here is a resistant isolate, which has a much smaller zone of inhibition, indicating that the cells can grow at higher concentrations of the drug. So resistance by definition is concentration dependent as it's measured by the MIC. Okay. So in addition, resistance in general, the way we think about it, and I think for the most part, this is the case, both in bacteria and in fungi, if you take a particular isolate and you just simply plate it on both a no drug and a drug plate, and then you got one colony that grew on the drug plate because it's drug resistant, then when you pick that drug resistant colony to a new plate and plate a similar number of cells, you get all the cells that land on the plate are drug resistant. And that's because this is a genetic property that's heritable and usually due to mutations that have been acquired, either vertically or very often in the case of bacteria horizontally, where mutations are passed from by plasmids from cell to cell. That doesn't happen in fungi. So in fungi, you need to acquire mutations if that's the way that you're going to become resistant. Now, mechanisms of drug resistance are also classic, and they're also similar in bacteria and in fungi. In general, excuse me, mechanisms of drug resistance are mechanisms that perturb the interaction between the drug and the drug target. So for example, if it's an enzyme, the drug binds the enzyme and inhibits its activity. And a resistant isolate may have acquired a mutation such that in the drug target, such that the drug can no longer bind or can no longer bind effectively enough to inhibit the activity. The other major mechanisms of drug resistance are drug inactivation. There may be a gene that, for example, in the case of the beta-lactam family, drugs that inactivate the genes that can inactivate the drugs. And there's also failure to activate drugs that are given as prodrugs. So they need to be activated, and mutations in genes that activate them would cause resistance. Another major mechanism is in changes in intracellular drug concentration, where if you can reduce the import or more frequently increase the export and efflux of drugs such that the intracellular drug concentration is low, that will make the cell suffer far less of the consequences of having the drug in its environment. So these resistance mechanisms are directly affecting the drug-target interactions and or the amount of drug that can have an interaction with the target. Now, the routes to acquiring drug resistance can be genetic and classically are. They're point mutations, insertion deletions, and of course also aneuploidy, which is what Anna Selmecki found when she was a graduate student in my lab oh so long ago. And what Anna found was a particular aneuploidy called isochromosome 5L, where there are 2 extra copies of the left arm of chromosome 5 in a palindrome flanking a centromere. And they have 2 important genes relevant for drug resistance on them. And these isolates, I want to just highlight, are resistant. So the parent is susceptible. And here's the strain that now has acquired the isochromosome 5L. Its halo is much smaller. It is resistant. And the mechanism here is very clear, and Anna has shown this through a number of genetic manipulations. ERG11 is the target of the drug, and if you have much more of it, you will have less effective drug concentration relative to its target. And TAC1 is a transcriptional activator of the expression of efflux pump genes, and it 2 extra copies, and especially homozygosis and 2 extra copies of it will generate, of an active form of it, will generate the ability to get a lot of the drug out of the cells. So extra copies of 2 different genes on chromosome 5L are enough to give you more targets and more efflux and give you increased resistance. Now, antifungal drug resistance, therefore, is concentration dependent due to changes in the drug-target interactions and its genetics with changes in DNA, either point mutations, which are classic, or aneuploidies, which can be transient. Or a newer report from Rob Todd and Anna Selmecki has shown that you can have smaller regions that are amplified in a mechanism that involves stepwise recombinations. That's really fascinating, and I encourage you all to see this paper. It's really quite exciting. So these kinds of copy number variations also can be transient, probably due to different recombination effects and so forth, but all of them are at the level of the DNA, and at least while they're there, the cells can be resistant. You can also get transposon mutations, and I'm not aware of having seen that, but that doesn't mean that they couldn't exist. And I think much of it is that we just haven't looked hard enough. The question, however, is really, does tolerance contribute to treatment failure? And so in order to start asking about tolerance, we need to define tolerance. And I'm going to give you the research definition, which is that tolerance is the ability of a subpopulation of cells in a susceptible strain to grow in supra-MIC drug concentrations. And that growth is usually slower than the kind of growth you would see from a resistant strain or from that strain in the absence of drug. So here I'm just showing you a few strains that have low, moderate, and higher tolerance on these disc assays. You can see that basically the halo size is the same. We're looking at them at 48 hours. At 24 hours, you would see the clear halo in all 3. And the halo, I guess you'll have to believe me, in this case is the same size. This is with fluconazole. This is E-test strips, which are just drug-impregnated gradients of drug that give you a halo and with low tolerance to caspofungin and much higher tolerance to caspofungin being shown here, but with very similar levels of resistance, that is MIC on the amount of drug that inhibits ultimately. And here we're looking at flucytosine, and it also can exhibit tolerance. Tolerance is detectable thus with different drugs, and I'm not showing it you here, but we have also shown it in different Candida species. Now, every strain has its own tolerance, and when Alon Dahan was in the lab, he did a large-scale survey of many, many strains, and you can see here just some samples of very low to very high tolerance strains. And in the 260-some strains that he analyzed, he saw a broad range of tolerance levels. We've even seen tolerance level that approaches 100% or 98%, things like that. So what this tells us is that strains with different genetic backgrounds have different tolerance levels. And what that means is there's a genetic contribution to tolerance. Okay, and it also tells us that if you want to understand tolerance, you better look at a lot of different strains. Otherwise, you'll only learn about the mechanism of tolerance that gives a particular level of tolerance in a particular strain. We don't know how many mechanisms there are, and we're suspecting that there's quite a few. I'll get into that later. Now, tolerance cells grow slower than resistance cells. I mentioned that normally we measure resistance at 24 hours and tolerance at 48. And a subpopulation response, the subpopulation response is not heritable. So when the cells are growing inside that halo, it's not everyone, it's a certain percentage of the cells. And if you take those cells that grew inside the zone of inhibition and the cells that grew outside the zone of inhibition, and you replate them on an identical assay, you will get identical results. So the cells that are growing as tolerant cells do not maintain that tolerant state. That is, the subpopulation response of the isogenic cells that you pick from inside and outside the halo is not heritable. It's due rather to a phenotypic difference between clonal cells. And so what I've just said to you, I know is confusing, and I'm going to go over it once more. You can have genetic differences between different isolates that have different tolerance levels because they have different genetic backgrounds that confer on them the ability to have different levels of tolerance under a given set of conditions. But when you have isogenic cells that are growing in drug, they have different abilities to grow in the drug. They exhibit phenotypic differences between the different cells in the population. Okay. I also just, so one other take home here is that tolerance involves a genetic component, which comes with the genetic background that you're looking at, and also physiological responses of the individual cells that are genetically identical within the population, but at any given time under a specific set of conditions, some of them will behave one way and some of them will behave the other way. Now, I just want to highlight that the routes to antifungal resistance and the routes to antifungal drug tolerance can be the same, and very often they are. In other words, there are point mutations or indels that could give rise to a change in the basal tolerance level of a particular strain. In addition, aneuploidy and probably gene amplification, although we haven't shown that directly, and maybe transposons, which none of us have checked yet as far as I know. If you have, please let me know, can give rise to tolerance or resistance, depending on what the change is. And I'm showing you here a really nice collection of strains that Feng Yang has collected in the lab over time. This is showing you just the ploidy level of the 8 chromosomes in Candida. If there's a color that is blue or purple, it's trisomic. In the case of these dark black parts, they're tetrasomic. And what we're looking at is the fact that he has managed to collect trisomies of either of 2 A's and a B or 2 B's and an A of the different homologs for essentially all of the chromosomes in Candida. And for chromosome 5, he also has monosomies. And almost all of these were collected using antifungal drugs and isolating different tolerant isolates. So tolerance is definitely something that can arise with aneuploidy. So phenotypic heterogeneity can certainly arise through genetic changes that change the stress responsiveness or the responsiveness to drug of the cells. Now, at the cellular level, I've mentioned this already, but I want to be really clear, tolerance is a subpopulation response. So tolerance is where different cells in the population, and these cells are sitting on drug, grow to different degrees. So you can see that there's some guys here who are doing a lot of growing, and there's this one who's doing very little growing. This is called a microcolony assay. It was developed by Levy, Zeev, and Siegel. And luckily for us, Naomi Zeev came and spent a summer in my lab and helped us set up this system so that we can track large numbers of individual cells and how much they're dividing. And she did this experiment with Alex Rosenberg. So you can see that different cells are growing and not growing, or growing to different degrees. And this is, again, a phenotypic heterogeneity of isogenic pathogenic cells. So if we took one of these clusters and probably plated them again, we'd see both kinds of responses. Now, I just want to highlight that this is reminiscent of many things that have been documented in Saccharomyces cerevisiae studies. So, for example, Audrey Gash's group in 2017 did a single-cell RNA-seq experiment of single cells before and after being exposed to a stress. So every column here is a different cell, and every row is a different gene. And she's looking at genes in the environmental stress response and ribosomal protein genes and ribosome biosynthesis genes. And what you can see is that most of the cells behave the same, both before stress and after stress, but in the population, and here it's at about 1%, there's a cell here that's definitely not doing what everybody else is doing. And it's very easy to see that there's that different cell. Now, this also brings to mind the original story of Levy, Zeev, and Siegel, where using microcolony assays, they were able to show that there are genes that are expressed in cells that don't divide very well when they're in normal situations. And they, in this case, they've labeled that cell with a GFP marked TSL1, which is a triolosynthase subunit. And then when they give a heat shock stress, it's that cell, most of the cells die because they gave enough heat shock that it was something that killed dividing cells. But this little guy sitting over here goes up and grows up and forms a complete microcolony. This is, the idea here is that this one is providing a bet hedging advantage. That is, this cell is holding all its cards and waiting until there's some situation where it can have an advantage. So in this case, slow-growing cells, the very rare slow-growing cells with high levels of TSL1 survive the heat shock. And it may be that there are different reasons for this in different cells and intolerance and different other responses to drug, but we think this is very interesting. And now to come back to Candida, I want to highlight some work from the lab of Mike Hallett, who's been collaborating with Malcolm Whiteway at Concordia University, and they've done single-cell RNA-seq, and they've been looking at the expression patterns. And what I'm going to focus on, they used a lot of different stresses. I'm going to focus here on the fluconazole at 48 hours, which is in orange. And all the cells and the genes that those cells are expressing are clustered into two patterns, one here and one here, which is clearly biphasic. That is, there are two different populations of cells expressing different things. And here you can see that the response A, which is the one over here, has lots of stress response things that we've seen in different fluconazole responses in some populations. And here it's more what he calls response B, or the boring response, because they're making ribosomal proteins and all sorts of things that are just kind of the general stuff that cells need. So the idea here is that one of these two populations is providing the ability to grow in the presence of stress, presumably this one, and the other population can't do that. Okay, but clearly there are two distinct subpopulation expression patterns, which I was thrilled to see because it's very congruent with what we're seeing. The question is, why do some cells grow and other ones don't? And the answer comes in part by using a fluorescent azole tag that was developed by Micha Friedman's lab at Tel Aviv University, together with Rafael Banhamu, his student, and Mayan Bibi in my lab. And what we saw was that this azole localizes to the cells. It actually localized to mitochondria, and then we synthesized a new one that localizes to the ER, which is where the target of azoles resides. And one of the things we found was that retargeting it to where its target is, relocalizing it, gave a large increase in efficacy. So the take home here is that if you localize your drug to the right subcellular organelle, that will increase its efficacy. But we use those fluorescent drugs to then look at the intracellular drug concentration in cells with different levels of tolerance, from low to very high. And in this case, we were sorting by flow cytometry and differentiating between high, medium, and low levels of drug by an order of magnitude. And what you can see is that cells that are low tolerance have many more, a strain that's low tolerance has many more cells that have high intracellular drug concentration than do cells that have high tolerance. So if you're highly tolerant, many of your cells in the population have about 10-fold less drug. Okay, so why do only some cells have less drug? Well, this brings me to the work of Marcus Ralser and co-workers. We've been collaborating for about a year. And one of the things that they've done is establish in Cerevisiae, again, in the baker's yeast, synthetic self-establishing metabolic communities, which are communities of cells that have lots of different genotypes. And the way they did this is with a single cell that's carrying four, it's a cell that is oxytrophic for 4 nutritional markers, 3 amino acids and a nucleoside. And then they put 4 plasmids to cover those oxytrophies, and each plasmid also has a fluorescent marker on it. So as the cells grow, they lose plasmid and they lose different ones randomly. And so what ends up generating is a mixed population of all the 16 possible genotypes of this original parent cell. And this is what a colony actually looks like when you image it with fluorescence for all of those different markers. What they think is going on is that the different auxotrophs within this colony are growing by exchanging metabolites with one another. The Lu- cell gives ura to the ura- cell, and the His- cell gives Lu to the Lu- cell, and so forth. And that by working as a community, they can all grow. So within the community, they can see a particular percentage of each of these genotypes for His, Lu, ura, and Met, for example. And the important thing here, without going into the details, is that when they stress the cells, the proportion of cells of each type changes, but these communities grow where none of the individual auxotrophs would ever grow. So by sharing metabolites, they're actually becoming more stress responsive, with the idea again here that these metabolic interactions between the cells involve different cells consuming different metabolites, using them through all sorts of processes that are important, including stress responses. And they also produce some metabolites that they give to their friends. So by having cellular heterogeneity, just like phenotypic heterogeneity that we see, they get more robust stress responses. Okay. And finally, I just want to raise very quickly the idea of whether tolerance is relevant clinically. And so the answer right now is no, because the instructions for what to do when you have an e-test strip that looks like the one on the right is to ignore the lawn of microcolonies and read at 80% inhibition. I have kind of just a gut level biologist problem with that, because those cells are growing and fluconazole is only a static drug. But the current guidelines say that it's not clinically relevant. So should we ignore it? And the answer, on the one hand, the clinic is saying no, but a number of studies have now suggested that we might want to take a second look from the Arendrup lab, and together with the Sunglar lab. There's a nice study showing that very high trailing growth, which is the same essentially as tolerance, affects the survival of 3 animal models, 2 mouse models and a Galaria model infected with Candida tropicalis. And we've done 2 small-scale studies, one of them in collaboration with Jules Eni, and she used strains from Arnaldo Colombo in Brazil, where there were a matched set of strains, some from patients that had non-persistent infections, that is, 1 course of fluconazole solved the problem, another where there were persistent isolates, and so another course of drugs was needed. And we looked at the level of tolerance in these isolates prior to them having received any treatment at all. And what we found was a significant increase in the FOG level, in the tolerance level for all of the strains from the patients who had persistent infections. So this suggests that persistent candidemia may be associated with higher initial levels of tolerance. And finally, in a study that just came out this year in a collaboration with Ronen Ben-Ami and his MEDS fellow Tal Levinson, we looked at a retrospective study of candidemia in 44 patients, 40 of whom were susceptible. There were fairly high levels of mortality. And what we found was that high tolerance, but not high MIC, was significantly associated with mortality for patients that were treated with fluconazole within less than 24 hours after candidemia onset, which is the recommended treatment regime. So death from candidemia was associated with high levels of tolerance. And when I say high, I'm talking above 50% of the cells were growing. So finally, I don't have time to go into the details, but I wanted to just mention that there are many, many pathways that affect tolerance. So all of these processes here, from the TOR pathway, HSP90, calcineurin, sphingolipid biosynthesis, protein kinase C, all of them, when you inhibit them, you inhibit tolerance and not resistance, or at least mostly tolerance and not resistance. And furthermore, inhibitors that inhibit them can then increase the sidality of the combination of fluconazole plus that inhibitor. The question is why so many different responses are involved, and I think the answer is that this is not a linear problem. It's not just A goes to B goes to C, but there are a lot of things involved, and depending on the genetic background, different ones can have a bigger effect. So in summary, resistance is not the same as tolerance, and tolerance is dependent on stress responses and phenotypic heterogeneity. Different isolates have different tolerance levels, and different cells in an isogenic population behave differently. There's clearly a lot to learn about how these cells are responding to stress, and it's a whole lot more complicated than probably most of us would like to think about. But that's what biology is. Sometimes it's more messy than we'd like, and that's what makes it fun to study. So with that, I'll thank you. I'll again acknowledge the people who did the work. This was my group at a lab meeting not long ago, and most people were there, but many of the people who did the work that I'm talking about here have since moved on to their own positions. And I asterisk those names that I've highlighted here who have contributed significantly to this work. And finally, and I'd really like to thank Marcus Ralser, who's an incredible collaborator and who's now a collaborator on a new ERC grant that we'll be starting in March in my lab to work on the metabolic effects on stress responses and drug responses. And with that, thank you very much.
Judy, thank you for that wonderful presentation. You've got 250 people clapping in the background there, although you can't see or hear them. So thank you very much for that. We're going to move straight on to the next presentation. So thank you again. Please, just to remind you to please put down the name to whom your question is addressed, the name of the speaker, because Joe and I have to scroll through all these questions, and there's questions now starting to stream in, but people are forgetting to put the name on. So please do that. Okay, so let's move on to our next speaker. So it's a great pleasure for me to introduce very briefly Michail Lionakis. Michail is a clinical academic who originally hails from Greece, who moved to the NIH in 2008 and then started his own laboratory at the NIH in 2012. What Michail does is he uses experimental systems to determine mechanisms underlying clinical phenotypes, and has made many significant contributions to our understanding of antifungal immunity. And we're going to hear about some of his recent insights today, which is going to tell us about why type 1 immunity is important for fungal infection control of the mucosa. Michail, over to you.
Thank you very much, Gordon, for this kind introduction, and also for having me on board in this, I think, phenomenal series about mycology, all aspects of mycology. So I'm an infectious disease and immunology trained physician scientist. So we study in our group how the host responds to fungal challenge. And what I'll try to do over the next 25 or so minutes is talk to you about a story we've been working over the last several years that hasn't been published yet, and for which we're very excited about. Before going into this, this is where we work. This is the NIH Clinical Center. This is the largest research hospital in the world, with about 300 beds, where we admit patients who have fungal problems in this particular case. And our labs are just next to it. And this close proximity and structure allows for bench to bedside investigations. And what I'll try to do today is give you a flavor of one of those examples that has had fruitful outcomes. So the program that we've developed here is interested in understanding host factors that are important against host defense against fungi. We focus on these two fungi primarily, but not exclusively, which collectively cause over 25,000 deaths in this country alone every year, despite medical therapy. And the way we've set up our program is multifaceted. So we take advantage of this unique place where we are, and we, over the last few years, enrolled over 500 patients who have inherited disorders in which these fungi are major players. At the same time, through the years, we've come to uncover and study certain iatrogenic risk factors. Some of them actually are phenocopies of genetic inhibitors, inhibitions, and we'll talk about that very, very briefly. At the same time, we do a lot of studies in clinically relevant mouse models of infection, and our approach is twofold. Either we start from human observations and we go to mice, try to understand the mechanisms, and ideally go back to humans, which is an example of what I'm going to talk about today, or the alternative, which we will not have time to discuss today, which is starting from basic studies in mice and then attempting to provide human relevance to those. So the goal of the lab overall is to understand the mechanisms by which we all respond to fungal challenge and develop better treatments and diagnostics for these patients who, as you heard from Judy, have poor outcomes. So today I'm going to only focus on Candida, just actually only mucosal candidiasis, and give you an example of an inherited disorder that has led us to an exciting new approach of how human fungal susceptibility might occur. So candidiasis doesn't need much of introduction, especially after Judy's great talk earlier, but it's important to note that it comes in two flavors. Most of us are colonized when perturbations in the immune system happen, or microbiota disease can happen. And it can happen in two flavors, either mucosal disease or systemic. And these are the same fungus, but they really are extremely different diseases when it comes to host defense. So you need lymphocytes to defend against mucosal disease, but phagocytes against systemic, and not vice versa. So for example, AIDS patients develop mucosal but not systemic candidiasis, and neutropenic patients develop systemic disease but not vaginal candidiasis. And that's an important distinction, and we'll get to that as we go through. Now, very, very briefly, systemic disease really relies on phagocytes. And this is a short video that shows in red a Candida albicans strain in a mouse kidney, which will filament, and then gray cells, which are neutrophils, will come very angrily and destroy it in a 2-hour time span in vivo. So neutrophils are absolutely important. Patients who lack neutrophils or their neutrophils don't function do not do very well when they are infected systemically with Candida. And in a single slide that I would like to highlight but not expand further, I want to draw everybody's attention to a very critical pathway that I think over the last decade has really catalyzed our understanding of human host defense against fungi. We're all familiar with toll-like receptors, and those in genetic models, either of MYD88 or IRF4 deficiency, these patients develop severe bacterial or viral disease, but not fungal disease. Why? Because there's a very important other pathway, the C-type lectin receptor pathway. Gordon discovered dectin-1 close to 20 years ago now, but there are dozens of those that recognize fungal carbohydrates. They signal through a kinase called SYK, and downstream of that, there is CAR9. Why is this an important pathway? Because CAR9 deficiency is the only immune deficiency that we know to date over the over 400 plus that we have in our armamentarium, in which that only fungal infection susceptibility happens without any other infections and no other non-infectious complications. You heard from Rebecca Drummond earlier in August in one of the MycoTalks how these patients get horrible fungal disease, either in their brains, because of the absence of recruitment of neutrophils in the tissue. So this is an immunodeficiency that causes tissue-specific neutropenia, if you will, but also causes severe skin infections. And why is this an important pathway? Obviously, the specificity to fungal diseases is critically important. We only understand a few things from a few of those receptors, so a lot more to be known. SYK inhibitors now are all over the clinical practice in diseases actually that already are susceptible themselves to fungal disease. So, and there are already reports of severe fungal infections in these patients. Actually, the most recent applications of SYK inhibitors is in severe COVID-19 to ameliorate hyperinflammation. And as we've built up our recruitment, we've now have enrolled over 150 patients who have severe skin and CNS fungal disease. Actually, 90% of them do not have CAR9 deficiency, which has opened up a fascinating window of discovering either upstream or downstream or parallel pathways and genes that actually confer tissue-specific, fungal-specific susceptibility. So more to come in the future, but it's an important pathway for systemic fungal disease in these sites. But today we'll talk about mucosal disease. And over a decade ago, the important contribution of IL-17 signaling in this specific form of candidiasis was well recognized. This is seminal work from Sarah Gaffen's work group, where Heather Conti had shown that if you look at fungal burden in the tongue of mice, well, wild-type mice control it, but mice lacking critical components of the IL-17 receptor signaling, they don't. 2 years later, followed by critically important papers in humans, showing that humans that have null mutations in IL-17 receptor or its adapter downstream, they don't signal through IL-17 receptor signaling. They develop chronic mucocutaneous candidiasis, that is refractory mucosal fungal disease. They also get skin staphylococcal and bacterial lung disease infections, so pointing towards a mucosal barrier problem. Now, fast forward now, there are several biologics, again, the theme of a iatrogenic susceptibility that models genetic inherited susceptibility, and a lot of those biologics do result in mucosal candidiasis. Now, how does this work? Again, back from Sarah's work, there's dependence on IL-17 signaling for production of potent antimicrobial peptides that are produced in the mucosa to defend against Candida. These are a couple of them. There are defensins also. And that's in mice, but also in humans, where you can see if a patient treated with IL-17 inhibitors, over 80–90% inhibition of the signaling. Now, what is absolutely important to go as background before we go into the actual data is that this is an extremely robust axis that protects all of us from mucosal fungal disease, despite most of us being colonized. 2 important points to make. So a very deep decline in IL-17 signaling is actually necessary for infection. As I showed you in this slide, over 85–90% of inhibition of signaling with these novel inhibitors only results in candidiasis in about 2 to 3% of patients. So you need sustained and very deep decline in order to get in trouble. In agreement with that, 50% decrease in signaling in either mice or in humans, and that is relatives of those patients who have null biallelic mutations do not get Candida. In addition and in parallel, there are several lymphoid cells that are actually produce IL-17 in the mucosa, predominantly CD4 T cells and gamma delta T cells, but also TC17 and ILC3s that can actually compensate for IL-17 production at the mucosa. So for example, if we look in mice, if you knock out alpha beta T cells or gamma delta T cells alone, these mice are not susceptible to Candida. If you get a RAG knockout and you cross both, then that you get in trouble. And in humans, actually, patients with idiopathic CD4 lymphocytopenia, that is lacking CD4 T cells without having HIV, AIDS, or more recently, rare patients who have absolutely no CD4 T cells because of null mutations, they're actually not susceptible to mucosal candidiasis, except for sporadic cases, while they're susceptible to other AIDS-defining classic infections. This raises the possibility that actually even in AIDS patients, the susceptibility is not only conferred by decline in CD4 T cells, but probably decline of IL-17 in other cell types. Actually, Pedro Lopez, who just joined our lab, is working in a macaque model now of SIV infection to actually clarify that. Now, this is important. Why? Because there are several other syndromes where we focus on, enrolling a lot of those here at NIH, and these are summarized here, that also manifest with chronic mucocutaneous candidiasis, but don't have mutations directly on IL-17. All of those have been attributed to having IL-17 deficiency on the basis of well-performed studies in the systemic circulation. For example, APOSIT patients, which we'll expand more later in the talk, a majority of them have autoantibodies that are neutralizing in the periphery against mostly IL-17F, but some against IL-17A. And then these 3 other diseases have been shown to have decreased numbers of IL-17 producing CD4 T cells in the blood. Our entire study and focus over the last few years has been on the basis that mucosal immunity has really not been examined in any of those conditions. And as I mentioned earlier, and you're all aware of, mucosal versus systemic disease are extremely different. So unless looking in the mucosa, it's very difficult to infer what the true mechanisms might be. So to do that, it's more difficult than want to do that. So over the last 7-8 years, we've developed at NIH 2 complementary approaches. One is a mucosal immunology program in humans, by which, in collaboration with Nikki Moutsopoulos at NIDCR, we get biopsies from patients who have these diseases to try to look at correlates of immunity at the site where it matters. And at the same time, working extensively in mice, where we phenocopied these syndromes in humans, as you see here with air deficiency, STAT1 gain of function, DAC8 deficiency, and Job syndrome. And what has come out of this work is that some of these diseases are actually 17 deficiencies, and some, surprisingly, are not. So focusing very briefly on an IL-17 deficiency disorder, this is DAC8 deficient mice that are susceptible to Candida. And as IL-17 receptor null mice are unable to upregulate any of those IL-17 dependent molecules, the mechanisms are not selective to Th17 cells, but rather broadly on all mucosal lymphoid sites at both the quantitative and the qualitative level. But let's really talk today about a disease that we now follow over 150 patients, which is by far the largest cohort of this autoimmune disease in the world. This stands for Autoimmune Polyendocrinopathy, Candidiasis-Ectodermal dystrophy. It's caused by mutations in a gene called AIR, or autoimmune regulator. And it's fascinating in the sense that it presents with a lot of autoimmunity and a single infectious disease. It was initially reported to cause predominantly hypoparathyroidism, Addison's, and Candida in a very classic characteristic triad. Through our work through the years, we've significantly expanding the scope of this to include over 30 different clinical manifestations, of which still only one is an infection, and that is chronic mucocutaneous candidiasis. A little bit about what this disease is. So AIR is a transcriptional regulator expressed in the thymus, where it protects all of us from actually developing autoimmunity by expressing in the thymic epithelium peripheral self-antigens and clonally deleting all of the self-reactive T cells that would encounter those self-antigens in the presence of AIR. So these patients that don't have that mechanism, what happens is self-reactive T cells escape in the periphery. They go to cognate tissues, they infiltrate them, they attack the self-antigen of choice in the cognate tissue, and they cause autoimmunity. And that's been shown to be the mechanism, as if you take AIR knockout T cells and put them in a mouse that doesn't have T cells, they develop autoimmunity. In addition, these mice and humans have autoantibodies, predominantly humans more than mice, but nonetheless. And at least in mice, serum transfers haven't shown these autoantibodies to be directly pathogenic. Now, in the setting of candidiasis, these patients, the majority of them carry neutralizing autoantibodies against Th17 cytokines, as I mentioned earlier, predominantly IL-17F. So our hypothesis through the years has been that these patients develop Candida in the mucosa because they have problems in their local mucosa-specific immune responses. To test this hypothesis, we generated the first model of oral candidiasis in these mice, which, as opposed to wild-type animals that control the infection, are unable to control the fungal burden in their tissues and their mucosa. Instead, again, in the same genetic background, if you inject Candida systemically, these mice actually are more resistant, highlighting again the site of infection matters, and immunity is context and tissue specific. In addition, phenocopying the human experience, these mice are not susceptible to systemic or oral viral infections, to skin staphylococcal disease, GI, Citrobacter, or ligature periodontitis. These three red represent models that depend on IL-17 competency for control. So this is a model that has mucosa, fungal specific susceptibility, phenocopying the human disease. In the histology, what you see is after 5 days, control of the fungus in the wild type setting, and an epithelial destruction from infiltration with Candida, as shown here, despite the presence of an immune response that is apparently not effective. And we'll talk about that right now. So this is a slide that summarizes work over 4 or 5 years that led us to the conclusion that not only the susceptibility is not caused by autoantibody-dependent neutralization of IL-17 signaling at the mucosa, but instead, and more so, that these mice and patients actually do not have mucosal IL-17 deficiency. Very briefly going through the data, actually the mice are susceptible, although only about 10% of them carry these autoantibodies. Extensive serum transfers into wild-type mice does not promote susceptibility to candidiasis. More importantly, if you look at IL-17/22 production by all relevant mucosal lymphoid cells in both their deficient mice and humans, are completely intact. So this is IL-17A, F22 by the 4 relevant immune cell types in mice after infection, normal. And this is human oral mucosal lymphoid cells. Again, the production of these cytokines are normal. Downstream of IL-17, there are all of these antimicrobial peptides, again, defined by Sarah Gaffen's group initially. And all of those, again, are intact. So let me remind you what happens if you don't have IL-17 receptor signaling. You don't induce those. In the setting of air deficiency, this induction is not only normal. In several of them, there is increased production. These are now mice. If we go to human mucosas from air deficient patients, S100A9 at the mRNA level in both gingival and buccal mucosa is intact, as is the protein at the level of the saliva. Now, this is just one readout, S100A9. We went through the process of doing this in an unbiased manner, that is with using RNA sequencing. And on the left, it is oral epithelial cells from primary oral epithelial cells from the tongue of mice, not an easy task to do. On the right is actually oral mucosal biopsies from healthy volunteers versus APICET patients. And what was concluded from this was in agreement with that I showed you earlier, whether looking at IL-17 induced or repressed genes against curated through Sarah's work, none of those were really impaired in the setting of APICET. So that led us to try to understand potential alternative mechanisms of susceptibility. This was very surprising to us. So we started by looking at the temporal and spatial accumulation of all leukocytes using flow cytometry and immunofluorescence to summarize the defects being here. So there was a marked increase in the accumulation of CD4 and CD8 T cells that I'm not showing you here for the interest of time in the air knockout oral mucosa, both before and throughout after infection. These were highly activated effectors with high expression of CD5. And where they accumulated were, as opposed to the wild type uninfected setting where, okay, there are 3 CD4s here, there are a significant accumulation in the intraepithelial and submucosal areas, more CD4s than CD8s, as Flo also confirmed. In humans, the same thing applied. That is, if you take a healthy oral mucosa, there's no CD4 or 8s. This is an uninfected APICET patient that has a lot of CD4s and CD8s in the intraepithelial and submucosal mucosa of the mouth. Now, if you look at these images in both the mouse and the patients, this is exactly how air deficient other tissues affected by autoimmunity look like. If you look at the lung, the salivary glands, the endocrine tissues, there is excess of lymphocytes. As I mentioned earlier, these are self-reactive T cells that can get into the mucosa. So this started looking like potentially another autoimmune disease, perhaps driven by T cells. So to show that and examine that, we took 2 approaches. To eventually show that pathogenic TCR alpha beta cells are actually both necessary and sufficient to drive susceptibility in these mice. So on this panel, what you see is that you have a wild type and air knockout mouse. The knockout mice are susceptible. If you cross the air knockout mouse with a TCR alpha beta knockout, that is, if you eliminate those T cells from air deficient mice, these mice control fungus. Now, we know T cells being protective in the setting of fungal disease and particularly mucosal candidiasis. In this particular autoimmune setting, they're actually pathogenic. They're not only necessary, but sufficient. Why? Because if you take air knockout T cells and you adoptively transfer them in a mouse that doesn't have T cells, that's sufficient to cause susceptibility. So this points to a paradigm by which actually T cells are detrimental in this particular setting. So what do these make? What do they do? What they make is a lot of interferon gamma. That's now data at the total oral mucosa level, a significant induction of interferon gamma even before Candida and throughout candidiasis, and interferon gamma inducible CXCL9 at levels almost as much as 0.5 micrograms per gram of tissue. This is specific for type 1 immune readouts as opposed to type 2 Treg and type 17. And in humans, if you look in saliva of these patients, there's a significant increase of CXCL9 in their mucosa. And the gamma is coming from both CD4 and CD8 T cells. This is now mouse data, oral CD4 lymphoid cells, CD4 T cells and CD8s. And the same thing with oral mucosal T cells from patients. CD4s and 8s make a lot of interferon gamma. So we now have a model by which T cells are detrimental, and what they appear to be making selectively is an excess of interferon gamma, which in the setting of the intestinal barrier, an IBD, has been well established to be detrimental for the mucosal barrier by promoting epithelial death. It's anti-proliferative, and it impairs epithelial permeability. So we consider these as potential effectors in this setting. So we focused on the epithelium to show that, again, in oral epithelial primary served from the tongue, there is a significant increase in phosphostat-1 that is all gamma-driven, because if you have an air gamma double knockout mouse, this signal is gone. So this is type 2 and not type 1 or 3 interferon-driven process. And in humans also, you see a lot of phosphostat-1 in the mucosa. And these are uninfected individuals. This says what we now call these disease features a type 2-driven mucosal interferonopathy. Now, we concluded that by looking at the T cell byproducts. We then took a 30,000-foot approach by looking at whether the excess interferon gamma was actually a dominant pathway. And again, looking at RNA sequencing here, it's in oral epithelial cells from mice. By far and by very, very impressive p-values, interferon gamma pathway was by far the most predominant in mice, but also in humans. As you see here, again, interferon gamma being the predominant response in oral epithelial tissue. We actually done this now in about 15 patients, both in gingiva and buccal mucosas. The data look the same. And if you source IPA in both the mouse and the human data sets, interferon gamma and STAT1 are the two predominant pathway modules, hubs of enrichment. So now we've established that there is a lot of T cells that make a lot of interferon gamma, and the interferon gamma is actually dominant in both mouse and humans with these, in this setting. So we examine whether those epithelial barriers were abnormal, and indeed they were. Now this was all that development of new assays. There's not much of literature about how to do this in the oral mucosa of mice and the tongue. This is work by Jigar Desai, who employed intravital confocal to show in situ that there is a lot of more epithelial dead cells by staining with PI and Hurst. And this is a gamma-driven approach, because if you eliminate gamma from this setting, the epithelium is restored. The same thing with proliferation. It's anti-proliferation in the setting of air deficiency, restored if you don't have gamma. I'm not showing you that for the interest of time. And then Vasikonomou devised a fitsidextran equivalent of what we do for IBD work, but now only in the tongue, to show that indeed air deficiency impairs the oral mucosal barrier. And this is driven by the excess interferon gamma, and in association with that, clotting defects, which would go with that. So that told us that gamma is necessary to drive this. Then we turned to Mark Swidergirl and Scott Filler, who used human oral epithelial cells and showed that actually excess prolonged interferon gamma is sufficient to also impair the oral mucosal barrier. As shown here, in a dose-dependent manner, gamma leads to epithelial death. It impairs permeability to the levels as if you added Triton X in the monocultures, in the monolayers, and it impairs tight junction. So interferon gamma is sufficient and necessary to impair the oral mucosal barrier. Does it make a difference in the fungal susceptibility? And the answer is yes. If you genetically eliminate interferon gamma in air deficient mice, the fungus is controlled and the mucosal injury is restored. So this same data we can now show with two FDA-approved modalities, a monoclonal antibody against interferon gamma and a JAK inhibitor that obviously has exciting translational implications, as I'll briefly mention in my conclusion slide. Then we went back to these adoptive transfer experiments to show that if you have air knockout T cells. I showed you that earlier. You can actually drive the phenotype. But if you take away the gamma production by these T cells, this susceptibility is gone. That says that gamma production by air knockout T cells is the driver of the disease. So what I've shown you here is that in addition to the well-established paradigm by which interferon IL-17 is absolutely critical for protection against this infection, fungal disease at the mucosa can also happen not as a problem of resistance, but immunopathology. In this particular setting, excess interferon gamma is the driver. And in this cartoon, what you see in the setting of air deficiency, where you have autoreactive T cells escaping, those make a lot of interferon gamma in the mucosa in the setting of normal IL-17 axis that drives defects in the epithelial barrier that allows for Candida to exploit the local niche and cause susceptibility. So where are we heading? What that drives really the activation and skewing of these cells? We've excluded the role of microbiota using germ-free mice. We favor self-antigens, and we have preliminary data pointing to that. And we're very keen in identifying those because these would have implications, I think, beyond APICID in other settings. Using conditional knockout mice, we want to pinpoint this to the thymus, in addition using thymic transplants, but there's now air expression in other cells. We're very interested in defining the molecular mechanisms of how excess gamma impairs the oral barrier. Why? Because a lot of those molecules are actually, again, druggable, and they could have translational implications. A major infection is excess interferon gamma sufficient to drive mucosal fungal susceptibility. The answer is yes, using mice that are at steady state transgenic, overexpressing interferon gamma, they are susceptible to disease. Could this microenvironment actually affect Candida virulence? I think that is likely based on some preliminary data, and using transcriptomic approaches, we want to define that more carefully. We've now started looking in other diseases that we have already data to implicate the same axis being relevant. I'm listing a few of them, but there are others. And the obvious question here is whether JAK modulation could ameliorate candidiasis and even broader autoimmunity in these patients. And we've already treated patients with this, and we're very, very excited with the results that we've gotten. So what I've shown you today is that IL-17 receptor-mediated antimicrobial peptide production is crucial for protection during mucosal candidiasis. Actually, I'm just summarizing a bulk of work from Sarah and other groups. But although T cells are protective and contribute to 17 production during mucosal candidiasis, what I'm showing you is that in certain settings, aberrant maladaptive T cell responses can also promote fungal infection at the mucosa. And the mechanism appears to depend on excess gamma STAT1 signaling that has direct damaging effects on the epithelial barrier and promote mucosal candidiasis. And in data that I don't have time to expand, but I certainly showed you a flavor of this disease, which is a devastating autoimmune disease, is a type 2 interferonopathy that can be targeted therapeutically. There's only one type 2 interferonopathy known in humans, HLH, and APICID, I think, is number 2. With that, I will end by summarizing what we aim for in the lab. By understanding basic mechanisms in the mouse, we want to eventually develop personalized approaches to re-stratify, prognosticate, and treat patients. By uncovering iatrogenic risk factors of fungal susceptibility, we could develop targeted treatment and prevention approaches. Again, as we see these patients who have devastating infections, developing novel mechanism-based treatments for disease is what we strive for. Now, this is my most important slide here. I will acknowledge the people that did the work on the project that I showed you today. This is Vasilis Economou and Tim Brake, who really spent close to 6-7 years completing this, what I showed you today. And Jigar Desai, who really developed the assays with intravital confocal microscopy. And then Greg, Elise, Monica, and Tom on the clinical recruitment and treatments. And with that, this wouldn't be possible with some key collaborators, Nikki Moutsopoulos, who I mentioned, Yasmin Belkade, Mark, and Scott, as well as the others. And with that, I would like to thank you for your attention. We'll be happy to take any questions.
Thank you very much, Michalis. Jules, could you just stop my camera, please? Thank you. Michalis, fabulous talk. Another 250 people giving you an applause in the background. Thank you. Thank you very much. So we'll move to the question and answer session straight away now. Thank you. And thank you for all your submitted questions. As I mentioned, we're going to tag team with Joe, and I will tag team between reading out questions from for each of the speakers. And I'll hand over to Joe, who will start with the first questions for Judy.
So I want to begin by thanking Judy and Michalis for really scintillating, thought-provoking talks of basic and clinical importance. We have about 30 questions that have come in so far. So I'm going to group some of the questions together. And the first one, Judy, centers on what drives the heterogeneity in the tolerant communities. Do you think this is stochastic? Are there genetic and epigenetic components, and maybe involving the transcriptome or the epitranscriptome as well?
Okay, so the answer to that would be we don't know, but yes. In other words, we haven't examined it carefully enough, but we think that all of those things could be contributing to it, and that they may be contributing upstream and downstream. So you can envision all sorts of interactions between different players, such that the status of the cell influences the epigenetics status, which then influences who gets expressed. And so you could have an awful lot of feedback in the process. And so that's that's the only answer I can give right now. And basically we haven't gotten deeply enough into it to say anything more.
Well, it will be fascinating to figure it all out. The second question I have is, are there mechanisms that allow the tolerance to be reset in the cell? And is that part of the process whereby you always have heterogeneity? And I'm wondering if it might be something analogous to aneuploid colonies that lose the chromosome and go back to euploidy. Is there something like that that resets the tolerance mechanism?
Right. So we're really fascinated by this question of how long you have memory of a particular state, and also how much the memory, so basically you could think of any tolerant cell has a progeny cell, and if that progeny cell inherits, some of the same messages, proteins, metabolites, everything that's involved, it would be more likely than the neighbor who's doing something else to be like that. But at the same time, there does seem to be some reset in the sense that even if you pick out the cells that were growing better in the drug, they'll still do the same thing over again when you replate them. One of the things we don't know, and this was raised in one of the questions, is whether this is because of cell-cell contact, because obviously when we pick them off the plate, we have to disperse them to plate them again. But there's lots of things that could be affecting it. And the answer again is no, but we don't know, but yes, all of those things are possibilities. I think we're at a stage where there's lots of interesting ideas, and we're hoping to start nailing them down in the not too distant future.
Great. I'm going to hand it over to Gordon to take a few questions from Michalis, and we'll tag team back and forth.
Thank you. Michalis, so there's been a couple of questions about the T cell specificity of these pathogenic T cells, and whether I know you mentioned in your last slide that this was work that was ongoing, but whether you knew whether there were antigen specific for Canada and possibly other commensals?
Yeah. So I do not have a conclusive answer to that question, but I can tell you what we are looking at. So that has, I think, two major kind of directions where we're looking at. So one is whether they're Candida specific T cells driving this. I think it's unlikely on the basis that all of those defects that I showed you are actually before Candida comes into play. So uninfected mice that you all know are not colonized with Candida as commensal, they already carry these defects. So whether Candida specificity exacerbates that at the second level, it is a possibility, but I don't think that that's a driver. The specificity of T cells, and we have started tackling that by doing single cell RNA sequencing and single cell TCR sequencing of mucosal T cells from both mice, and we're moving to humans, is to see whether there is an oligoclonal repertoire in the mucosa, and that's our prediction, and whether we could potentially, using that approach, gain more insights with regards to what could be the self-antigen specificities that we have data to suggest that are actually displayed in an air dependent manner in the thymus that allow then for air deficient T cells to go out, find them in the mucosa and cause the barrier dysfunction, even in the absence of Candida. So we're working on those two directions, and I think they would be critically important to help us understand how this works.
Okay, so related to that is the germ-free mice, whether you've tested that and whether they have the same pathology or not?
Yes, so we've made the air knockout germ-free mice, and they do get a lot of interferon gamma, and they are still susceptible to the infection. And we've also used the AM&V cocktail in air knockout mice, and we had the same result. So it appears that it is not directly related to the microbiota, although, you know, germ-free mice just eliminate everything. So, you know, whether they are competing colonizers and commensals that might have effects, it is not 100% ruled out.
Thanks, Michail. Last question before I hand you back to Joe. So there's a comment here that inhaled gamma is well tolerated in humans, and so your model suggests that you're getting epithelial damage. So the question is whether you think the toxic effect is on the basal cells?
So I'm aware of systemic gamma intermittently. Inhaled gamma, I must say, you know, it is used in some settings. I don't know if it is used on a daily basis. So one consideration is whether, and we have data in the transgenic mice for interferon gamma, whether you absolutely need a certain threshold of both extent of exposure and time of exposure. So whether inhaled interferon gamma, the way it's used in humans intermittently, is sufficient to cause the phenotype in the respiratory epithelium, I do not know. Certainly these patients who get, you know, 3 times a week systemic gamma for CGD, for example, they never get oral candidiasis. So I think there are time and dose dependent effects. And whether the basal cells specifically are affected, our data would suggest that it is not necessarily only the basal, but also on the apical. So, but more work would need to be done in the respiratory epithelium, where not much has been looked at there.
Thanks, Michail. Back to you, Joe.
Judy, we have a question for you about how the mixed communities share resources. Does this involve direct contact or diffusion or vesicles? And related to that is, which do you think comes first, tolerance or the metabolic adaptation?
Okay, so first of all, in terms of how they share, we know that there's lots of extracellular metabolites that are found in the milieu around the cells or in the liquid medium if we wash them in liquid medium. So certainly there's a lot of export and import of metabolites. Now, whether they are naked or whether sometimes they're in extracellular vesicles, we don't know at this point. There's some evidence from Cryptococcus that extracellular vesicles could be involved, and some work in Candida on extracellular vesicles, although not in the context of drug resistance, suggests that they could in parallel be involved as well. So that's certainly not a route we can rule out. We can't rule out anything because we haven't done enough to look at that, but I certainly think that it could be by multiple routes because there's multiple metabolites being shared as well. In terms of the second question, which was...
which comes first, do you think?
Oh, which comes first? So again, we don't really know, but I think that Marcus, who's the metabolite guru in our team, would say that it's an intrinsic property of metabolism. And so it's not a which comes first, but rather all of this is going together. So, I mean, what we think is probably going on is that as metabolites get shared and sharing becomes important in these, at least in the synthetic communities and probably in the Candida albicans as well, that's what we're going to be testing, you would have increases in exchange across and between the cells, and that would start creating new states, and then those states would somewhat establish but not be super stable. And so it's something that is kind of intrinsic to the metabolism itself. There's always stuff going in and out of the cells. And the idea here is just that that has a lot more purpose than we've ever been thinking about it, especially at least those of us who think mostly about DNA makes RNA makes proteins. So.
Great. And then the second question is, are species that have a high tolerance more prone to develop resistance to the drug they're tolerant to compared to other drugs? In essence, is tolerance a sort of stepping stone to resistance?
Okay, so that's a great question. And in bacteria, there's some work that says, yes, it is. In bacteria, the tolerance is a little bit different, but, you know, sort of intuitively, one would think that if you have a higher effective population size because you're dividing somewhat, then you, then you by definition have a higher chance of developing mutations, some of which would be beneficial mutations. So I think that's very likely, but I don't think it's something where A goes to B goes to C in a linear thing. It's just that there's a better chance of it. Yeah.
Great.
Sorry about my guard dog. Back to you, Gordon.
Thanks, Joe. Actually, that's great because that's the first dog we've had speaking in MycoTalks. It's great. So your dog makes the first. Michail, back to the epithelial damage. The two unrelated questions, but around that subject area. The first question is around about macrophages. So macrophages, obviously, we know respond very well to gamma driving inos, etc. Is there a role for these in the epithelial damage that you see?
We have not yet formally tested that, but our hypothesis is that yes. So there are actually increased numbers of macrophages, and they do exhibit features of activation. So what we would need to do, and we just haven't done that yet, is to either eliminate them or neutralize some of those pathways that could potentially mediate the damage. So I think that it is very likely that the T cells make the interferon gamma, and then the effectors are myeloid cells. We have focused on the T cells predominantly because the AR T cell double knockout mice didn't have the phenotype, but we are working on figuring out whether it's exclusively oxidative burst dependent or not. That, you know, that's also an important point to clarify. But yes, great question.
Okay, and then the other question that's related to tissue damage is in your patients. I mean, the damage to the mucosa. I mean, does that influence susceptibility to other pathogens? Why is it specific for Candida?
Yeah, so in patients, the mucosal infection susceptibility, I would say the infection susceptibility in general is confined to mucosal candidiasis, and the same thing with humans. So that raises, I think, an important question, which is, I think, what you're inferring and looking here in the Q&A box, which is why Candida specificity in the setting of a hypergamma driven epithelial barrier defect. So if we look at the other two infections that can happen in the oral mucosa, viral and bacterial, for viruses, we do have experimental data that actually having a lot of gamma is protective. And I think we know that from how interferon gamma is critical for intracellular host control of viruses. So if you eliminate the interferon gamma from these mice, you actually get, that's when you start getting viral susceptibility. So I think the viral absence of viral infections likely relates to how interferon gamma is actually promoting protection. Now, for bacteria, it's less clear. Now, you know, there are not that many bacterial oral infections actually that happen in humans, but there are a couple of areas where we've started investigating. So one possibility is that some of these chemokines downstream of gamma, such as CXCL9 and 10, actually have very potent antibacterial activity, but not antifungal activity. And that's been shown at levels where way lower than actually the levels in the human saliva and the mouse mucosa. So there is a possibility that bacteria are controlled through effectors that come through that pathway. The alternative or parallel explanation is that there's something very specific about the microenvironment that is driven by gamma that is specific to Candida because it enriches the ability of Candida to invade, adhere, and or increase virulence. And that's an area where we've started accumulating some data, and I think that is a plausibility. If you take, for example, a Candida strain that is not able to filament, the ability to infect an air knockout mucosa is significantly impaired, almost diminished. So I think there might be fungal attributes on its own, like filamentation, and triggered by interferon gamma excess that might allow for the Candida specificity in combination with factors that relate to excess gamma that might be protective against viral and bacterial pathogens, if I can summarize it in one sentence.
Thanks, Michail. Back to you, Joe.
Judy, there are a couple of questions about tolerance and relationship with the host. And one of these is, does tolerance impact pathogenesis in the absence of an antifungal drug? And are there host factors that impact tolerance? You're muted.
Okay, we don't know. There just hasn't been enough measurement of tolerance in the field to be able to talk about most of those things. So we don't know about pathogenicity, for example, in different strains. I mean, one of the goals of our grant is to collect 1,000 different isolates of Candida albicans from across the diversity. And for those that we get patient information about the site of infection, the outcome of the infection, that would be great. But the goal is to then characterize all these different kinds of phenotypic aspects, as well as the genome, the proteome, and the metabolome of these isolates, so that we can actually start to generate those correlations. So stay tuned.
Great. There's a related question asking what is known about strain heterogeneity at different infection sites, maybe the skin versus the mouth versus the kidney, for example.
So about tolerance and how it might be different from different sites. So far, we have analyzed several hundred strains for tolerance and have looked at whether there's any correlation with their site of infection, and neither the site of infection nor the country that it came from nor any other thing that information that we had about those strains was correlated with them.
Okay, thank you. Back to you, Gordon.
Thanks, Joe. There's been several questions come in, Michail, about the impact of this air-induced damage and its impact and susceptibility to other mucosal sites, gut, vagina, and others. Would you care to perhaps discuss what you've discovered in those areas?
Yeah, so what we've looked at in the air deficient setting is only two mucocutaneous sites, the oral mucosa and the skin. And the susceptibility is not is only the oral mucosa, not the skin. Actually, 8% patients don't get skin candidiasis. We have not looked in the GI tract, predominantly because in humans, GI candidiasis is really not that much of a an obvious clinical entity, but it's something that would be worth looking. And we have not yet examined vaginal disease. I would say that the mucosa where the predominance of susceptibility occurs in humans is the oral mucosa, actually not so much of vaginal candidiasis, although we see some. So whether it is specific to oral versus vaginal and GI and what are the regulators of that, we need to find out. But in the skin, there is no susceptibility. Okay,
and then one quick more question before I hand you back. So the question here is something that is beyond candidiasis. When you target the interferon gamma stat signaling in these patients, do you see any other symptoms of this APEC, however you pronounce it, disease that are mediated in these models?
So we actually can cure the mice by inhibiting interferon gamma. So
I'm sorry?
We can cure the mice by inhibiting interferon gamma at the genetic level or with a JAK inhibitor, with universal survival as opposed to universal mortality. All tissues that have autoimmunity do not display autoimmunity. And we've actually treated now a few patients with a JAK inhibitor, and we've been very pleased with the reversal of all of what you would expect to be reversible autoimmune manifestations, from alopecia to enteritis to gastritis to hypothyroidism to Candida. So it would appear, that's what my last kind of conclusion there, that this is actually an interferon gamma driven autoimmune disease, not exclusively at the fungal infection level, but overall. And that is exciting because this gives us opportunities, as we've started doing, and we're preparing a formal clinical trial to actually put these patients in remission using a mechanism-based approach.
Thank you, Alice. I think we've got time for one more question. Joe, would you like to ask Judy one more question and then we'll close the meeting?
Sure. There was a question about tolerance versus heteroresistance, and is there an accepted difference by which you could classify isolates as one or the other?
Right, so heteroresistance has generally been a clinical term that describes strains that have a fairly, that at least initially have a fairly rare occurrence of isolates that grow in the presence of the drug. And then in most cases, those isolates will continue to grow for some time, much more like the epigenetic, the epimutations that you described last week, Joe. The distinction that we've made, because we do some work with heteroresistance in Candida glabrata, where we see it much more frequently. In albicans with fluconazole, at least, tolerance is really dominant, and we wouldn't be able to see heteroresistance on top of it. Heteroresistance is rare. It's usually 1 in 1,000 cells or something like that, in that range, and not 1% or 10% or 90%. So we think that the mechanisms have to be pretty different when you have it rare versus frequent. And on top of that, many times the heteroresistant isolates then have the ability to maintain their state. So it seems like at least a transient heritable state, whereas with tolerance, it seems less so.
Fantastic. Thanks so much.
Yeah.
Well, we've run out of time, and I'm aware that we haven't got through all the questions. So thank you very much to the attendees for submitting all these fantastic questions. It's really been a very nicely interactive session. So thank you. And I want to once again thank Michail and Judy for really fantastic talks today. It's really, Judy, you were worried about keeping the stand up. You've absolutely done it, both of you. Thank you very much. And from behalf of myself and my co-chair, Joe, thanks to everyone. Have a good break if you're having one, and we will see you again in 2021.
Okay. Thank you.
Bye.
Bye-bye.